Hello, I'm Thomas Klein, and this is my presentation on the hourglass model, which is one model for conservation during embryonic development. First, an introduction into the field of biology, which studies the evolution of developmental processes. This field is known colloquially as Evo-Devo and studies how developmental mechanisms have evolved in different organisms. This field provides a unique approach to studying homologies because it investigates the processes at the root of morphological diversity. The beginnings of the field can be traced back to Darwin, who postulated that similarities in embryos implied common ancestry. It wasn't until much later that the advent of genetic techniques such as sequencing has allowed for investigating the molecular mechanism behind development. One such discovery is that of homeotic genes, which control the development of body structures. Some of these genes show examples of deep homology, which is the idea that structures with morphological divergence still have conserved developmental genes and regulatory circuits. These processes can be so conserved that they still functionally work the same way in very different organisms. One example is that cnidarian developmental genes induce the formation of sensory organs in certain arthropods. Here we see an example of this in a fly. This presentation will focus on the phylotypic stage. The phylotypic stage is a stage of embryonic development in which those members of a phylum show the most similarity to each other. Early investigations into the similarity of embryos began with morphological studies. One early definition of the phylotypic stage is shown here. It reads, "The phylotypic stage is the point in development where embryos exhibit the defining morphological features of their phylum before diverging into a more specialized form." While this is a useful definition, you may remember that seemingly disparate morphologies, such as those found between cnidarians and flies, can still be controlled by conserved processes. Again, an example of deep homology. Now, the shift is focused away from structural changes and instead begin to focus on molecular techniques which show similarities in the biochemistry of these organisms. Different models of the evolutionary conservation of development place the phylotypic stage at different points throughout embryonic development. Carl von Beer, considered the father of embryology, believed that the most conservation of traits between species could be found in the early stages of embryogenesis, with increasing divergence as time progressed. This theory, known as the funnel model, can be seen pictured all the way to the right of the slide. Later, Ernst Haeckel coined the famous term ontogeny recapitulates phylogeny, which hypothesized that embryonic development mimics the evolutionary history of an organism. A diagram of one such example is seen to the left of the funnel model diagram. A recapitulation may look something like a mammal embryo, initially resembling an aquatic creature, then a reptile, and finally a mammal, reflecting its evolutionary history. The final model we'll discuss is the hourglass model. This model proposes that the most conservation is observed in a mid-developmental stage, with increased divergence between organisms in early development and late development. One important milestone in the synthesis of the hourglass model was the discovery of Hox genes. Hox genes were discovered in Drosophila flies as the regulators of body segment development. They are responsible for determining the body plan of organisms. Mutations in these genes were found to cause the development of ectopic structures, or structures that develop in abnormal locations on the body. For example, some mutants had an extra wing develop where a balancing organ should have been found. Homologs of these genes were discovered in vertebrates, and studies expressing Hox genes across species showed functional equivalency, meaning that they produced homologous structures despite being in different organisms. Hox genes are of a similar class and control the development of specific organs. The picture on the right shows a study that expressed the eyeless gene from mice in Drosophila flies, which cause the formation of ectopic eyes. These genes are expressed during the organogenesis phase of embryonic development, which is a mid-developmental phase. Therefore, the presence of these genes supports the hourglass model. Another important advancement in the hourglass model was the invention of next-generation sequencing. The developmental process is marked by an abundance of transcription factor cascades. These cascades occur when the activation of one or a few genes begin a chain of events that result in the expression of many genes. For example, Hox genes encode transcription factors that induce the expression of other transcription factors, inducing yet more genes in a cascade comprised of multiple levels of induction. These cascades create many RNA transcripts that encode the proteins which drive development. The transcriptome is the collection of RNA expressed from these genes, and it essentially reflects what part of the genome an organism is utilizing at any one time. Next-generation sequencing allows for the characterization and quantification of the many RNA transcripts created from such a cascade, which gives us insight into what genes are being used during development. Many transcriptomic studies of sequence similarity and levels of expression show the most similarity in the middle stages of development, supporting the hourglass model. Gene sequence studies evaluate the similarity of information encoded in genes by comparing orthologous genes. Orthologous genes are found in species that share a common ancestor with that gene. After speciation, orthologous genes diverge, but if the shared function remains necessary, divergence is limited and therefore conserved. The first study listed here, from Hazkani-Kovo et al., showed that the most similarity between mice and humans was found in a mid-developmental stage of embryogenesis. Another type of transcriptomic study is a gene expression study, which compares the relative amounts of similar RNA sequences produced between organisms. The second and third study here used mice, frogs, and zebrafish, and 6 different Drosophila flies respectively, to discover that RNA expression during mid-embryogenesis is the most similar. A different type of study, which tries more to focus on the function of genes, is a gene age study. Gene age studies show similar results, with the oldest, most conserved genes being expressed during mid-embryogenesis. Gene age studies evaluate when a gene first emerged in evolutionary history. Genes found in early organisms can be foundational to more diversified morphologies and are conserved as a result. Gene age studies use a technique called phylostratigraphy to discover what the most recent evolutionary ancestor of 2 species is. Genes which are found in a more distant ancestor are older genes, and therefore represent a more conserved function. The 2 studies listed here used zebrafish to find that the oldest genes were expressed during mid-development. The second study found a similar result in flies and plants. Notably, conservation across plants and animals is especially remarkable due to their distant evolutionary relationship. Their most recent ancestor would have been a single-celled organism, meaning that each developed embryogenesis independently. Therefore, the embryonic mechanism conservation is an example of convergent evolution and shows that this mid-developmental phase has very important functional relevance. Although there is a large body of evidence supporting the hourglass model, much evidence supporting the funnel model directly refutes those findings. This first study found in a gene sequence similarity test that the highest sequence similarity was found in early development, directly refuting a study we had mentioned earlier. These results were consistent with the findings of the second study from Rue, who showed that the genes expressed early in zebrafish and mouse development have the most severe effect on the development when knocked out or removed from the genome. Importantly, both of these studies use vertebrate models, and both of them point out that much of the corroborating findings for the hourglass model come from Drosophila models and do not hold up in vertebrates. Again, another one of the previously referenced studies supporting the hourglass model was not able to be validated in anything besides flies. A unique method of analysis was undertaken by Comte et al. In this final study. They measured changes in interactions of regulating proteins and RNA, and thus were able to compare the similarities of regulation mechanisms in zebrafish and mice. They found conserved regulation profiles during mid-embryogenesis, but noted no difference between this and later stages. This led them to propose that the apparent similarity observed in other data is an expression of conservation found earlier in development. In conclusion, early morphological studies and more recent molecular studies have confirmed the existence of conserved stages of embryonic development, but trends in conservation are convoluted. Embryology has benefited greatly from the new level of inquiry that molecular techniques provide. For example, the discovery of the ubiquitous function of Hox genes and similar body plans genes provides evidence for the conservation of the organogenesis phase, a mid-developmental phase, therefore supporting the hourglass model. However, the mosaic of information that new techniques provide creates difficulties in decrypting conservation. Transcriptomic analysis seems to provide evidence for both the hourglass and funnel models, but with different respects. Results supporting the hourglass model seem to show a strong similarity in the transcriptome during mid-development. However, results supporting the funnel model suggest that the observed similarity in the middle stages is caused by genetic regulation triggered earlier in development. This conclusion allows for both models to explain a different aspect of developmental evolution, and would argue that the strongest evolutionary constraints are placed on the foundational mechanisms present in early development, that then give rise to similar mid-developmental stages. So, while the majority of data do suggest that the most similar stage of development occurs in mid-embryogenesis, aka the hourglass model, the cause of this conserved stage may be found in early development, aka the funnel model. Here is a list of references used both on screen and in my commentary.